We said that nucleic acids contain two types or examples of that particular chemical type. That's DNA and RNA. Now, the acronym DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So one of the differences between those two forms of nucleic acid is the particular sugar. In DNA, the sugar is called deoxyribose. In RNA, the sugar is called ribose. Now, each of these sugars are 5-carbon sugars, and attached to one of these sugars, you'll have other components. And one of those components is a nitrogenous base, and perhaps you've heard of in DNA the bases are designated by a single letter, A, T, G, and C. We'll look at more detail about those in just a moment, but those are examples of the type of base that might be attached to the deoxyribose molecule in DNA nucleotides. There is a difference between DNA and RNA nucleotides, and we'll look at that in a little more detail later on. Now, this is a simplified version of the sugar, and at each one of these points is where a carbon atom is. So we said that DNA contains deoxyribose. Deoxyribose and ribose are both 5-carbon sugars. So this would be position of carbon number 1. This is position of carbon number 2. And that carbon is represented by the symbolism 2', carbon 3, 3', carbon 4 would be 4', and this upper carbon that we see here is the 5' carbon. So you'll notice that deoxyribose, D means to take away, oxy referring to oxygen. So one of the differences chemically between ribose and deoxyribose is that deoxyribose lacks an oxygen in the 2' position, meaning that there's one less oxygen chemically in its makeup. Now, to make DNA and RNA, we have to join nucleotides together. And this 3' carbon with its OH group attached is going to be critically important in explaining how new nucleotides get added on. So when they talk about the reactive tail, they're talking about the OH group that's present on both ribose and deoxyribose that's attached to the 3' carbon. And again, we'll come back and look at another figure in a few moments that shows how these individual nucleotides hook together to make the long chains of nucleotides observed in deoxyribose and ribose.